This lecture covers innate and adaptive immunity. There are two main types of immune responses: a non-specific immune response, also called innate immunity or natural immunity, which acts as a first line of defense and offers resistance to any kind of microbe or foreign material, so it's not specific. And one key feature of innate or non-specific immunity is it lacks immunological memory. So upon reinfection by that particular pathogen, there's no specifically novel thing that happens, unlike with adaptive immunity or antibody-based immunity. So one important arm of the innate immune response is complement. Complement proteins are basically just small proteins synthesized in the liver. They circulate in the blood as inactive precursors, but then become active through a variety of mechanisms, as pictured here, one of which is the lectin pathway, another the classical pathway, and then the alternative pathway. In the lectin pathway pictured here, these proteins that bind to carbohydrates called lectins will coat a microbe, and in that process will activate a particular serine protease, which performs the downstream functions. In the classical pathway, a complement protein called C1 interacts with antibodies that have already bound to the surface of a microbe, and that cross-linkage will turn this into the serine protease that will in turn process proteins downstream to perform the next step. And then in the alternative pathway, one of the complement proteins, C3b, will coat a microbe, specifically looking for glycoproteins on the surface of a microbe. And when this happens, that feeds into this next step, the formation of C3 convertase. C3 convertase is the product of C2, complement C4 being cut into two parts. One of those parts, C4b, comes together with C2a to make this key enzyme, C3 convertase. So C3 convertase can be seen here cutting complement C3 into C3a and C3b. So C3b acts as an opsonin, essentially. It coats microbes and enhances the ability of phagocytic cells to eat that particular microbe. But C3b can also bind to C3 convertase to make the next enzyme in the cascade. C3a, pictured here, acts as a chemoattractant that can actually enhance the immune response, but it can also lead to inflammation. So the formation of C5 convertase performs the next step. So C5 is then cleaved by C5 convertase to make C5b and C5a. So this is another chemoattractant, again, to draw immune system cells to the fight. And then C5b can group together with C6, C7, C8, and C9 to form what's called a membrane attack complex, which can punch its way into bacterial membranes or viral envelopes in order to create pores. So here are those three pathways in the context of a SARS CoV2 infection. Here's the classical pathway, and imagine SARS CoV2 now coated with antibodies, and then the C1 protein binding to those antibodies, cross-linking, and then forming a serine protease that then cleaves C4 and C2 in order to make these intermediates that ultimately are used to make the C3 convertase enzyme. Here's a mannose-binding lectin that sticks to the spike proteins of SARS-CoV-2, recognizing them as glycoproteins. And basically that activates a membrane serine protease that can also cleave the complement proteins to ultimately make C3 convertase. And lastly, the alternative pathway is considered a tick-over event in which C3 is hydrolyzed ultimately into C3b, but C3 here has to bind to factor B and then factor D, and basically factor B here is cleaved by factor D, and in turn this complex forms the C3 convertase as well. And so in this process of forming C3 convertase, the C3 convertase can then cut C3 into C3a, the chemoattractant, and even more C3b. And so then C3b can interact with C4b and C2a, basically the C3 convertase, to form the C5 convertase. Once the C5 is made, it can cut C5s and produce more chemoattractant, which again contributes to inflammation, and then the formation of C5b, which can stimulate the formation of the membrane attack complex. This can attack viral envelopes, but it can also attack virus-infected cells or even non-virus infected cells of our body, and that can cause some serious problems. And I purposely left out this event over here, just showing you how these feed into the formation of C3 convertase and C5 convertase. So in this panel here, you can see SARS-CoV-2 binding to ACE2 receptor, activating complement, and ultimately activating each of the three pathways, which in turn results in the formation of C5 convertase, which cuts C5 and then produces the chemoattractant C5a as well as C5b, the first component of the membrane attack complex. So C5a can essentially act as a chemoattractant to macrophages and monocytes, and PMNs as well, polymorphonuclear neutrophils, that can come to the site of the infection and start fighting the infection. This infection ultimately triggers the formation of cytokines such as interleukin 8 and MCP from the endothelial cells. So imagine these are endothelial cells that line blood vessels, for example. And so these chemoattractants like MCP that you see here will attract even more of these immune system cells to the fight. The production of this membrane attack complex that punches holes in this membrane also triggers the formation of these receptors, E-selectin, for example, and ICAM, that will further alert these immune system cells where specifically to bind in preparation for penetrating the endothelial cells so they can fight an infection deeper in the tissue. So this recruits these cell types to push through and then ultimately cause damage to this endothelial tissue, endothelial lining. These stimulated endothelial cells will in turn produce a variety of reactive oxygen species, peroxides, and reactive nitrogen species, and so forth, that ultimately will cause damage to the surrounding areas and further add to the damage and inflammation. As more and more of these immune system cells push their way through the endothelium, that in turn will cause leakage. And if this happens to be a capillary or blood vessel, as fluid leaks out, that will cause severe drops in blood pressure. Another hallmark of the SARS-CoV-2 infections, and even from vaccination, is the formation of blood clots. And so in this particular case, MAC formation in these endothelial cells can lead to the exocytosis of von Willebrand factor and P-selectin. So you see these molecules that build up on the cell surface and that cause platelets to aggregate, and this leads to that coagulopathy that I mentioned. This in turn also leads to release of thrombomodulin from the cell surface. Thrombomodulin shedding in turn encourages the conversion of fibrinogen to fibrin, and as a result more clotting can take place. And here you can see a membrane attack complex that is formed in a platelet membrane, which causes it to leak additional coagulation factors. So all of this can lead to blockage and in turn vascular injury and extensive blood clotting. A powerful innate immune defense is the production of interferon by infected cells, as you see here. So this is a virus infection taking place. The virus releases its genetic information, and as the virus replicates its genetic information, it transiently produces double-stranded RNAs that alert the system here, and transcription factors such as NF-kappa B, to activate the expression of interferon. Interferon in turn will be produced and secreted, and the neighboring cells that are not yet infected will be alerted to the presence of this RNA virus. In particular, the interferon will stimulate the formation of the expression rather of RNase. The RNases will then attack RNAs from invading viruses and degrade them. But another thing that it does is activates protein kinase R, which can phosphorylate initiation factors and therefore modulate or dampen translation or protein synthesis in this cell. And by dampening protein synthesis, viral proteins cannot be synthesized quickly enough. One of the interesting features of SARS CoV2 is that it produces a non-structural protein called NSP16 that appears to methylate RNA and decrease its recognition to the rest of the cell, and therefore essentially cloaking itself. Another component of the innate immune response are natural killer cells. And as you can see from these images, natural killer cells are designed to kill intracellularly infected cells. They're essentially looking for the wrong combination of receptors on the surface, in particular the loss of or the diminished expression of MHC class I molecules on the surface of virus-infected cells. If an NK cell detects that unusual aspect of an infected cell, it will pump that cell full of perforin and granzymes. Perforins are membrane punching, hole punching enzymes, and granzymes are proteases. These would come into a vacuole, and the vacuole membrane would be destroyed by the perforins, which would then punch holes in nuclear envelope and mitochondrial membranes, for example, while the granzyme proteases would destroy essential proteins inside the cytoplasm, thereby causing apoptosis or programmed cell death. Here are some ways in which the NK cells can also recognize and target infected cells for destruction. So in this particular case, the NK cell is actually looking for antibodies on the surface of an infected cell. One hallmark of an infected cell, especially if it's infected with a virus, is that the cell will display virus antigens on the surface. And when this happens, those antigens will attract antibodies, very specific antibodies to those antigens. And this combination will allow the NK cell to also recognize this as an infected cell and then to pump this cell full of perforin and granzymes to destroy it. So here's this NK cell concept in the context of a SARS CoV-2 infection. This is a low-risk individual, a healthy individual with a healthy population of natural killer cells. Here's an airway epithelial cell infected with SARS CoV-2. The epithelial cell is displaying antigens, and again, these can be coated with antibodies. This cell would also have a reduced display of MHC class I antigens as well. Either of those cases would trigger these NK cells to attack it, as you see here. So the NK cell is attacking this cell, destroying it with perforin and granzyme, but then also communicating with other cells of the immune response to alert them and attract them to the site of the infection, resulting in a recovery. A high-risk individual, however, has a population of dysfunctional NK cells, and by dysfunctional typically mean that there's a decreased expression of granzyme production, for example, so less of that serine protease. And as a result, these dysfunctional NK cells can bind and try, but they can't clear the infection. And so the virus proliferates, and that triggers these NK cells to produce even more cytokines to do, very minimal work. So more immune system cells come to the fight and produce even more cytokines, but the virus infection doesn't clear, and so these things just magnify to the point where the NK cells themselves eventually become exhausted, and then the lungs are damaged extensively by all the chemicals that are produced during this infection. Another type of immune response then is the specific immune response. And so I talked about non-specific and now specific immune response. The specific immune response and non-specific immune response work together. They're not happening in parallel to one another. They're crossing over and working synergistically to one another. They recognize a specific foreign agent. That's why it's also called adaptive immunity, where the immune system is adapting to a particular pathogen and developing a heightened specific response. Another great thing about this type of memory this immunity is it has memory, meaning that its effectiveness increases upon repeated exposure. And as I said, they work together. So one of the main players are B cells that mature in the bone marrow. They circulate in the blood, they can settle in lymphoid organs, such as the spleen, for example, and after maturation and activation, they can become plasma cells that produce antibodies. Another player in the specific immune response are the T lymphocytes that mature in the thymus, which is in your chest, basically. They can remain in the thymus, circulate in the blood, or reside in lymphoid tissue. And like B cells, they require antigen binding to a surface receptor for activation. They produce chemicals that have an effect on other cells, particularly cytokines, that can then activate and recruit additional cell types to the fight. And then this is just showing you the lineage, starting with a lymphocyte stem cell, that if it migrates out of its location and into, in the bone marrow, for example, and receives the right stimulation, will become a B cell. If it migrates out of the bone marrow and into the bloodstream and ultimately into the thymus, it can become a T cell. And then in the case of the B cell, B cells can differentiate into antibody producers and memory cells, and T cells as well. T cells can actually differentiate into helper cells that coordinate the immune response, cytotoxic T cells that destroy intracellularly infected cells, just like NK cells, but these are specific to specific pathogens. And they can also form memory. So the specific immune response begins with phagocytosis, which is basically different cell types, such as tissue macrophages or monocytes or dendritic cells or neutrophils, actually recognizing and ingesting and killing extracellular microbes. The mechanism of recognition looks something like this, where it's a combination of both innate and specific defenses. In this case, you see a phagocytic cell with its antibody receptor binding to a microbe that's been coated with antibody. The degree of binding is not that strong. The opsonin in this case is the antibody. So again, opsonin is a term for any molecule that increases the ability of something to be eaten or phagocytosed. In this particular case, you see the phagocytic cell with its C3b receptor binding to a microbe that has C3b complement coated on its surface. And then this binding's a little tighter. But by far, having both of those events take place, the antibody and C3b binding, is synergistically a more enhanced degree of binding. And so this cell would then be destined to be ingested and destroyed, as pictured here. So the antigen is ingested, broken into bits, and then bits of that microbe are displayed on the surface using MHC, either class 1 or class 2 MHC antigen. So the first step in this cascade, after a phagocytic cell has ingested antigen and is now displaying it, is interaction with a T cell, specifically the T cell receptor on either a CD4 T helper cell or a CD8 cytotoxic T cell. And so if it happens to be a CD8 cytotoxic T cell that recognizes this CD8 T cell would then go off and kill any cells that are infected by whatever produced this antigen. But in the case of a CD4 T cell, this handshake that takes place here will stimulate the production of additional immune system cells and also act as a chemoattractant to bring more of them into the fight. One of the interesting aspects of this is this T cell receptor has already recognized this antigen, even though it may not have ever seen it before. And the reason for that is that there's just a huge population of T cells with roughly 50 to 100 million different T cell receptors. So basically a key to fit any lock. And so the right T cell must recognize the antigen and be stimulated. And so with respect to SARS CoV2, it looks something like this. Here's our antigen presenting cell, and then these dark green objects are bits of SARS CoV2 from any part of the genome, any part of the ultrastructure, any proteins from the ultrastructure of the virus. So here's an infected cell, and this infected cell is now displaying bits of SARS-CoV-2 antigen, and some of these antigens are leaking out and basically being phagocytosed by the phagocytic cell pictured here. Now this phagocytic cell is also ingesting viruses and destroying them as well. So not only is this infected cell priming the phagocytic cell, but the phagocytic cell is also ingesting viruses and priming itself. And so here you see the phagocytic cell presenting antigen to a population of T cells, but only the right T cell with the right T cell receptor will go on to stimulate a defense, basically. Meanwhile, here's and so this individual APC also will present to CD8 T cells, the cytotoxic T cells, which can then go on and recognize any infected cell that's displaying those same antigens, just like this was. And then that will target this particular infected cell for destruction. So this CD8 T cell will pump this thing full of toxins and destroy it. And so again, you see granzyme B, for example, as you've already heard, like with the NK cells, pumping it full of those toxins and destroying them. In either case, though, this can contribute potentially to lung immunopathology if you have an unregulated amount of these things being producing an unregulated amount of antiviral activity and various cytokines and so forth. Tumor necrosis factor alpha, for example, will attract more immune system cells to the fight. And if that's not carefully regulated, too many of them can come and cause tissue damage. So once the correct T cell has been stimulated, it can go over and search out the correct B cell for antibody production. And that's pictured here. So here's the CD4 T helper cell finding the appropriate B cell. And so the B cell in this particular case is acting as an antigen presenting cell. It has receptors on its surface to recognize those bits of SARS-CoV-2 antigen, and then will display those on its surface in conjunction with MHC class 2 as pictured here. So again, this T cell receptor, which has the right confirmation for binding to the antigen, fits like a lock and key and will now stimulate the proliferation of these B cells. So they'll activate and proliferate. They'll turn into plasma cells, memory cells, and they'll produce, plasma cells that produce antibody and memory cells that will remember this infection for a period of time. So here's another look at that. Here's the B cell, and the B cell has, as I said, a B cell receptor that happens to recognize the antigen on a bacterium or a virus or whatever. It will take that in, destroy it, and then display it on its surface. And so in this particular case, the B cell, the activated B cell now displaying antigen, can then be recognized by the appropriate T cell that's already been primed. And so in this process, the B cell can then be stimulated to divide into memory cells and antibody-producing plasma cells. And so these antibodies can fight the current infection, whereas these memory cells can remember the infection and protect you more quickly upon subsequent infection with that particular pathogen. There's a process called clonal selection in this that is at play as well. Just like with the T cells, you don't want every T cell with every kind of T cell receptor coming to the fight. You only want the most appropriate. And so in this particular case, the B cell that has the most appropriate receptor for the particular antigen in question is the one that will be activated and proliferate into plasma and memory cells. And again, this particular B cell will be recognized by a particular T cell, which stimulates this, what's called clonal selection. So here's this process more in the context of SARS-CoV-2 again. Here's the virus, and it's being processed by so-called M cells in the gut tract. These M cells will process the antigen, display them, or send them over to a pocket macrophage that you see here in the digestive tract, and then this macrophage will display to a helper T cell, and then the appropriate helper T cell will pick the appropriate B cell in order to activate it. as you see here, to become memory and antibody-producing cells. So along the gut tract, these B cells will produce a class of antibody called secretory IgA, which will then go out into the mucosa and basically into the lumen of the digestive tract and bind to any antigens and target them for destruction. So an important point I want to make here at this particular junction is that because of all the ways in which the immune system can be stimulated, it's become pretty clear that SARS CoV-2 infection actually confers a greater level of immunity than the vaccines. And I'll show you that in a subsequent slide, but this has also led to a lot of controversy in terms of people refusing to get vaccinated because they've already had the natural infection. And the basis for that argument is along the lines of what I showed you, but also pictured here, where imagine the virus entering. There's all these different places in the upper respiratory tract and lower respiratory tract where a variety of immune responses take place. And so here in these tonsillar adenoids, for example, you have all the typical things, plasma cells that can produce IgA, IgM, and IgG, 3 classes of antibodies. The alpha beta T cells, and which, again, this are CD4, CD8 T cells, which can activate the immune response and kill intracellularly infected cells. All the macrophages, the antigen-presenting cells, and dendritic cells, and so forth. So there's a robust immune response here in these adenoids. And then as you get deeper, there's all these, again, these pockets where immune system cells reside to produce secretory IgG, as I just showed you with the gastrointestinal example. And then goblet cells producing mucus that can trap the microbes as well. But in all these different locations, as I said, the ability to produce immunoglobulin M to protect your immune response, to protect your respiratory and digestive tract. When you get a shot, you're getting the shot here intramuscularly in your shoulder, and so the immune response is relegated to the lymphatics and the bloodstream. So you're essentially just stimulating IgM. Well, initially IgM and then ultimately IgG, but you're not getting the full extent of the IgA production, the protection that would happen along the respiratory tract, for example. And as a result of that, you're still seeing individuals being reinfected and being permissive to transmitting the virus because there isn't sufficient protection built up here. It's all in here. So this slide is intended to show you the whole process. Here you see a SARS CoV-2 virion binding to a respiratory epithelial cell. So again, imagine this virus passing through the upper respiratory tract and down the trachea and into the lungs, passing by all the various immune responses, the adenoids, and so forth. The virus receptor binds to ACE2, and in combination with a serine protease and furin protease, this receptor is clipped, and that allows the virus ultimately to enter, undergo membrane fusion, release its plus-stranded RNA, which is then translated into proteins and ultimately transcribed into new genomes. As the virus assembles in these vesicles, the mature particles are then exocytosed into the environment. And so then hopefully the phagocytic cells, like this dendritic cell, would be here waiting for it to engulf it and present antigens to T helper cells or cytotoxic T cells. The cytotoxic T cells with the appropriate T cell receptor become activated and then can kill intracellularly infected cells. The T helper cells with the correct T cell receptor can go on to shake hands with B cells, stimulate them to divide into memory and plasma cells. And the plasma cells obviously produce antibodies to coat and neutralize the virus, with the memory B cells standing by, waiting for a subsequent reinfection. So this slide is intended to show you the kinetics of antibody production following infection. It does not include IgA, just IgM and IgG, the two main blood-borne antibodies. So here on the y-axis is antibody level and then days after infection. Here's the incubation period in which the person is infected here, and then some amount of time goes by where the individual is asymptomatic, not showing any symptoms. But eventually we hit a point where the individual starts to show signs of infection, such as a fever. And during this phase is typically when the first antibody is produced, called IgM. It's not as specific and therefore less neutralizing, and therefore IgM production rises and falls fairly rapidly, giving way to the production of the more specific and better neutralizing antibody called IgG, as you can see here. And then IgG will reach not only higher levels, but also last much longer, depending on the infection. So these numbers should not be considered hard and fast. They differ from person to person and from infection to infection. But they can generally last for a long time, usually many months. But then eventually they dissipate. But what happens upon reinfection at some later date, months or years later, the memory cells of the B cell response can be stimulated much more quickly, and as a result so the IgM response still happens, but it's very minimal. But the IgG response happens much quicker and much more intensely, and so therefore this memory response is absolutely vital in fighting a subsequent exposure to a particular pathogen. So knowledge of how the kinetics of immunoglobulin M and immunoglobulin G antibodies are produced, as shown in the previous graph, should help you properly interpret how these lateral flow assays work. These are designed to detect both IgM and IgG production as a result of a SARS CoV2 infection. And so in each of these cassettes, there's a little conjugation pad here, and the conjugation pad contains SARS CoV2 antigen bound to gold particles, and then a rabbit antibody also bound to gold particles. And here is where your blood sample would go from a finger prick, usually with a sterile lancet. And then a couple drops of blood are applied, and that would push any potential antibodies in your blood across the conjugation pad and pick up the antigen, SARS antigen gold and rabbit antibody gold going as it moves through. If there's any antibodies in your blood, they will attach to the SARS CoV2 antigens bound to gold here. And as they push their way out, the antibody and SARS antigen gold complexes will get captured by anti-human antibodies that are placed here and here. So these anti-human antibodies will just grab onto any human antibodies that they found. But it just so happens that these would be, if they're SARS-CoV-2 antibodies that are bound to the antigens that are bound in turn to gold, they would accumulate in these two spots here, whether they're IgM or IgG. Up here is a anti-rabbit antibody stuck to the filter, and so that's just an internal control. So the rabbit antibody bound to gold here would move all the way through all of this because it's a rabbit antibody, and therefore won't get stopped here or here, and will collect here. And so the band will illuminate where the gold particles are concentrated. And so again, a nice bright red line tells you that the control worked. Again, the rabbit antibody gold complexes were successfully pushed through this by the buffer and ended up where they were supposed to, getting trapped by the anti-rabbit antibodies here. So anywhere there's a nice bright red line, that's a positive, and you must have a positive in each of these control lanes for your cassette to be useful. If you have a bright red line here for G, that tells you that you're producing IgG only, and if you have a bright red line for M only, that tells you that you're producing IgM only. If you have both, then both antibodies are present. And so again, based on kinetics of the antibody, we can figure out the time course of the infection, essentially. So if we see this, for example, well, let's go to this one. If we see this, where the IgM is first, and then but there's no IgG, then that suggests that perhaps we're at an early stage of the infection here. If we see this with no IgM, that may tell us that we are at a somewhat perhaps later stage in the infection where there's still circulating antibodies but no IgM anymore. If both are present, then we may be at some closer crossover point here in the infection process. But in either of these cases, this, or this, the individual would be considered positive for SARS-CoV-2. I put this slide in here not to paint vaccine makers as evil, but more so to just put into perspective who exactly we're dealing with. Now, yes, they are producing a product that is highly beneficial, but these are also business people, and they also have a business agenda. And so Prevnar gives us a great example of what I'm referring to, the vaccine that's intended to prevent streptococcal infections. Now, strep pneumoniae infections are a major cause of death and illness worldwide. It's estimated that 1.6 million deaths were caused by this agent annually. That about 0.7 to 1 million of those are children under the age of 5, and these are obviously mostly children in poor countries. But when we look at the composition of this particular vaccine, Prevnar, it's primarily developed for the US and European epidemiological situation. In other words, it's formulated to prevent infections with strep pneumoniae strains that are mostly a problem in wealthy Western countries. And so again, I'm just saying this to remind everybody who we're dealing with here as I get into this conversation about vaccines. Now, there's a vaccine problem with most microbes, and influenza presents a really good example of that. Influenza undergoes what's called antigenic shift because it has a segmented genome. And so its genome pieces can enter the cytoplasm, and then these bits of RNA that make up the virus's genome can be swapped. And so that the result is a combination of progeny, some progeny that very closely resemble the parent strains, and then some progeny that are amalgams, essentially, chimeric versions of the parent strains. And so again, this new variant then could cause immune escape and present problems for vaccination. And this general concept that you see here is the reason why we have these really variable effectiveness rates when it comes to the flu vaccines every year. And again, it varies pretty significantly. I should also point out an important term here, the concept of efficacy versus effectiveness. Efficacy is more of an experimentally determined number in highly controlled trials, whereas effectiveness is how this thing works in the real world with all the messiness and sloppiness of the real world, where you're unable to control supply chain issues and cold chain issues and so forth. And I also want to preface any discussion of SARS-CoV-2 COVID-19 vaccines with what happened historically, especially with SARS-CoV-1 vaccines, where back in the early 2000s, the pharma industry invested hundreds of millions of dollars in SARS vaccine research, and then basically the virus went away. U.S. government and philanthropic agencies left these vaccine makers holding the bag, so to speak, and left them very weary about future investments in vaccine production. And so my point in bringing this up then is this probably is why there's a lot of cheerleading going on for everybody, regardless of your particular situation, to get one of these vaccines. But as I've shown you already with the headline from that science paper, having SARS-CoV-2 once confers much greater immunity than a vaccine. But still, they're not going to fully say don't bother getting vaccinated. They're going to still tell you, but vaccination remains vital. Of course it does, if you haven't been exposed before. And so that's one of the big issues, as I've pointed out before, this fight between natural immunity, those with natural immunity that have recovered from COVID-19, and people on the other side still pushing vaccination for everybody to the extent that you could lose your job or be sanctioned in other ways. So this is just a quick look at some vaccine ingredients. Obviously, there's an active ingredient, which is usually the piece of microbe, or in the case of the Moderna and Pfizer vaccines, the bit of messenger RNA that would encode the virus spike protein. So something that would stimulate the immune response directly. There's also adjuvants, which are included, that enhance or strengthen the immune response. Sometimes that's aluminum compounds that, all they do basically is slow the release of the vaccine components to facilitate a slower burn and a more optimal production of antibody over time. And they can be anti-caking too, just the same way that they use aluminum in pancake mixes and so forth. Preservatives, so that the vaccine is not contaminated. Often they've used controversial things like thimerosal, a mercury-based preservative, but it can also be straight up antibiotics or other compounds as well. And sometimes individuals are allergic to these compounds and therefore can't be given such vaccines. And obviously thimerosal, being mercury-based, has been very controversial, and therefore it's been removed from pretty much all vaccines in the United States, even though it's a very effective preservative and in super low concentrations. There's also emulsifiers for making sure everything stays together and doesn't precipitate. And then there's leftovers from production. So these can be any kind of preservatives like formaldehyde, glutaraldehyde, that are used to inactivate whole viruses in whole virus preparations, for example. Or even stuff left over from if viruses need to be cultivated. Now, this is obviously not a problem for the Pfizer, Moderna messenger RNA vaccines, where no live viruses are used in the process. But for other processes, for sure, this can be these carryovers can cause residual allergic reactions as well. So here's a look at the actual ingredients in the Moderna vaccine. So here's the prime antigen generator, the messenger RNA encoding the virus spike protein. So it's actually the cell translating this into proteins. That is the antigen generator. But there's also a variety of lipids. Obviously, you need a lipid coating in order to get that messenger RNA to move into the cells after injection. So all these lipids are necessary for vaccine delivery. And then, of course, there's buffers that keep the membrane stable and keep the RNA stable so that it doesn't degrade. And then other stabilizers, in this case, sucrose. So generally speaking, there's nothing super harmful in the vaccine ingredients, just normal buffers that are found in everyday foods. Sucrose, again, a sugar that's found in everyday foods, and lipids like cholesterol that are often found in everyday foods. And so again, nothing startling about any of these ingredients. There is potential for the spike protein itself to become toxic and stimulate an inappropriate immune response, which happens in rare cases, as we see with some of the vaccine-related injury that have taken place. So the overall process for getting a vaccine to market looks sort of like this. In this particular case, you've got the virus, and here's the virus spike protein. So now, in the case of the AstraZeneca vaccine, Oxford AstraZeneca vaccine, or even Johnson & Johnson vaccine is more along the lines of this, where they take an adenovirus shell and pack it full of DNA that encodes the SARS CoV2 spike protein. And then obviously the Moderna Pfizer vaccines are just messenger RNA fragments representing or encoding the spike protein. So in preclinical trials, you can just test these things out in animals to make sure that there's no obvious cytotoxicity. And then you can move to clinical trials once you're satisfied that they're safe. So phase 1 usually involves a few healthy individuals, just to check for safety in healthy individuals. And then you can move to larger groups of individuals and mix it up a little bit, so you can go across age demographics and so forth. Eventually you can scale these up to more global participation. The Pfizer, for example, had global trials in different countries and all across the United States. Moderna, I think, was mostly United States trials. So you want many cultural backgrounds, you want different age groups, you want different levels of risk, again, to people with preconditions and so forth. The idea is to be as representative as possible so that you can get a good assessment for the efficacy of the vaccine, and then ultimately it's mass produced and distributed, and that's when we get to find out the effectiveness of a vaccine. So this slide gives you an overview of the AstraZeneca-Oxford vaccines that were primarily used in the UK and the Johnson & Johnson vaccine. These are non-replicating viral vector vaccines that use an adenovirus shell. So this is a virus, a respiratory virus, but you're just using the shell, and inside it you put the SARS CoV2 spike gene and basically some other genes, but nothing that would allow this to replicate. So it's then injected, and the virus, pseudovirus essentially, is ingested, and then antigens are presented on phagocytic cells to stimulate an immune response. The great thing about these is that they only need refrigerator storage, which could be a problem in other countries that don't have deep freezers that are required for the other vaccines that I'll talk about. They have a pretty high efficacy from the clinical trials at preventing serious disease, not necessarily preventing infection, but preventing serious disease. So we'll see what happens with effectiveness. But the other great thing is that they're pretty cheap. And again, they're administered as a single dose, and that's why people are worried that you're probably going to need, well, not need, but you might require a booster shot if you're in a particularly vulnerable group. The RNA vaccines are the Pfizer-BioNTech and the Moderna vaccine. That's also co-produced by the NIAID, of which Fauci is the director. So the vaccine is a messenger RNA fragment that encodes the spike protein surrounded in these lipid nanoparticles that are then injected, and then the lipids allow them to undergo membrane fusion and insert. The viral proteins are translated, the viral RNA is translated into proteins, and then these are then recognized by phagocytic cells that ingest them and then display antigens to provoke an immune response. Now again, these are 2-dose regimens. They have to be ultra cold stored, which again, this cold chain issue is a big problem, especially in countries that don't have routine access to these deep freezers. The other problem with them is they're highly expensive, and again, not for most Western countries, but for many countries, this is not a very cost-effective approach. There's a slightly higher efficacy, again, at preventing serious illness, but is that efficacy the same for preventing infection? No. So another thing I like to point out is, just like with antibiotics, in which massive use of antibiotics, massive and inappropriate use of antibiotics, particularly antibiotics that are not completely sterilizing, and again used appropriately, can result in the formation of mutants that resist the antibiotics. And that's something similar can happen with massive use of vaccines as well. You get these sort of escape mutants that they're called, that form. Vaccines that do not provide sterilizing immunity and therefore continue to permit transmission will lead to a buildup of large standing populations of virus that greatly increase the risk of immune escape. The point is that the vaccine puts a really intense selective pressure on viruses to modify that particular spike protein target of the vaccine. And a lot of the modifications that take place are not detrimental to the virus, and so it doesn't really cost them much to make those changes. But what it can do is reduce the efficacy of the vaccines, and as a result, massive use of these vaccines can actually encourage resistance, which in turn would lead to a lot of resurgence in infections. So some of the lingering questions, are vaccine mandates and vaccine passports the right way to do this? What's the lasting immunity, or are we going to need boosters for the rest of our lives? How is this going to play out as well? Is there going to be long-term immunity? Will the memory response be robust? And again, will that lead to a desire for more boosters? What are the side effects? We really don't know. A lot of people have said, well, there's this thing called long COVID, so we shouldn't expose ourselves intentionally to this virus because we might have these unintended consequences. But the same could be said of the vaccines as well. What are the side effects? What's the true effectiveness? And how long will it take us to really understand what the true effectiveness is? Another big question, how does this end? Will we ever reach a state of herd immunity, or will the virus just keep making minor changes that will always result in escape mutants? Will it come from natural, vaccine, a combination of the two? Another one is, will there be natural attenuation, meaning the virus eventually just resolves itself to becoming transmissible like rhinoviruses that cause the common cold, but far less virulent as individuals just become more and more immune to it. And then, like a seasonal cold, can maybe kill the super frail, but will be less likely to kill the same types of individuals that it did at the start of the pandemic. And I give the example here of the 1919 influenza, which was an H1N1 that killed 50 to 100 million individuals worldwide. But in 2009, when we had the H1N1 pandemic, wasn't really a major killer. And I would actually argue that it was a major killer for younger people who hadn't been exposed to that. But for older individuals who had seen H1N1 previously, there was a lower proportion of killing, of death. So again, that sort of speaks to this concept that over time, individuals develop a significant memory, even if it's not perfect memory, even if it's memory to something related to it. That could be all that's necessary to avoid serious illness. Another big one is, maybe some really important antiviral. Now, there's several coming out right now, and there's lots of clinical trials on old stuff and some new stuff, molnupiravir and some of these other drugs. So we'll see how that goes. We're notoriously bad at our producing antivirals that are super effective at clearing virus infections. We have anti-HIV drugs, for example, but they seem to only be good at managing. They're not sterilizing. And then, of course, burnout. The virus can just smolder indefinitely, like we deal with influenza, like we deal with rhinoviruses and other respiratory pathogens year after year after year. And we just resume our lives. We cut this lockdown, masking, all these panicky maneuvers, and just go on living just like normal.